As chemistry needs flasks and test tubes, so biochemistry, the chemistry of life, has its own special containers. To make them, you simply add water to this waxy substance. In one of nature's more remarkable tricks, the chemical instantly forms itself into layers, membranes, too thin to see under an ordinary microscope. And the membrane layers spontaneously shape themselves into tiny bags, making a cloudy white liquid. So easy to make are these tiny chemical bags that something very like this process must have occurred right at the dawn of life on Earth. Evolution is based on such individual units. Each of these cells is enclosed in its membrane of this very same material. A cell is like a factory. On the site there's an office block with filing cabinets full of information, enough to define the whole living system of which the cell is part. 20 years ago, after a little breaking and entering, we learned to read the genetic codes hidden within them. The factory also has its manufacturing plant, its powerhouses, its warehouses and delivery systems, and its waste disposal systems. The spies from the outside world have learned a great deal about them too, but now they're paying a great deal more attention to the dividing walls inside and to the perimeter fence. Until a few years ago, this was the best picture of that outer membrane that could be obtained. As someone said, it looks like burned bacon rind. It's of the same material that we saw in those artificial bags, made out of this, an extract of cell membrane. It's a relative of soap. At the Agricultural Research Council's laboratories near Cambridge, Alec Bangham has studied its curious properties for many years. Here's how he demonstrates one of them. The trough now contains a mixture of warm water and ether. It gives off inflammable fumes. Let's see what a little soap can do to those flames.
Now here's a packet of soap powder, and let's see what it does when we put it onto water, and we shake it into water.
You're pouring out molecules that immediately stand upright in the water. Let's have a slow motion replay. One end of the molecule likes water. It's called hydrophilic, while the other end hates water. It's hydrophobic. So on the surface they all line up one way with just the hydrophilic head end in contact with the water. The tail is an insoluble fatty material. Surround the soapy molecules by water and they form tiny droplets. Oily dirt that also doesn't like water gets inside among the tails. Soaps and detergents form these droplets very easily because in water the molecule is conical in shape. They form a layer 1 molecule thick. The soap put out the fire because it stopped the ether escaping from the surface.
Now let's sweep away that monolayer of soap and reignite the surface and see what an extract of biological cell membranes will do. Jesus, burnt my fingers that time. That's it.
And this too has formed a monolayer. But now each molecule has 2 hydrophobic tails to each head. The head is usually based on a phosphate group, and here are the tails of fatty material, or lipid as it's called. The complete phospholipid molecule is much more substantial than the detergent. And when sitting on water, it has a cylindrical shape that forms a stronger barrier than detergent molecules do. That's a property of phospholipids that we depend on from the moment that we're born.
Is it coming? Right. Come on, nice big push down to your bottom for me.
As the baby emerges from the womb, its lungs are full of water. And yet, listen.
What a little boy!
The everyday miracle of a baby's first breath has been studied by Colin Morley.
The expansion of a newborn baby's lungs are a truly remarkable thing, because the lungs are full of watery liquid, and this has to be cleaved in order to provide an air surface. And it's very much like two plates of glass with water in between. It's extremely difficult to separate those. And in a similar way, it would be difficult for the baby to expand its lungs, but for the fact that the lungs contain a very special substance which lowers the surface tension and reduces the forces needed to separate the liquid in the lung and produce an air surface. We've studied this particular substance by washing out the lungs of dead animals, and you can see it's a frothy material that produces a very stable foam.
The phospholipids contained in this foam have another curious property. Here, a blade, suspended by a thread, rotates evenly in water. A few grains of phospholipid form the invisible monolayer on the surface. Now the sheet of molecules is squeezed to one end, and for a moment the surface becomes stiff. Now, and for the rest of his life, every time young Simon breathes out, that constantly renewed monolayer will be stiff enough to stop his lungs collapsing. That birth went according to nature's plan, but not all babies are so fortunate. Some, perhaps 1 in 100, don't have quite enough of the surface material to start with, and they have difficulty in breathing. Despite devoted, and for our medical services, time-consuming and costly care, in Britain alone, some 2,000 babies die every year. But if they can be helped through this bad patch, at its worst a few hours after birth, they'll develop quite normally as though nothing had happened. Premature babies, and those delivered by caesarean section, are at greatest risk that the natural phospholipid system may not be fully developed. So in the operating theatre, there's special equipment to help breathing. There, if it's needed, when the baby is born. The paediatrician is ready to move in fast. The baby's cry signals that already the main danger is past. But if it weren't, here's one possible remedy that may become available in the future. Colin Morley has developed a dry phospholipid dispenser. His powder is obtained not from the lungs of an animal, which might be contaminated in some way, but is a chemically identical extract from the yolk of an egg. In theory, it looks a good idea, but it is yet to be proved. If it does work well, that white powder could truly offer the breath of life to many thousands of children. These bubbles are another example of the same principle. But instead of a single layer of detergent molecules, we now have two, and a film of water between them. Bend that a little with air inside and out, and you have a structure like this. As water drains away, a soap film can become very thin. When it's comparable to the wavelength of light, the colors change with the thickness. Near the top, it's so thin that the colors have all gone. Thinner than the shortest optical wavelength, the bubble is turning black. The bilayer forming that flimsy soap film has its hydrophobic tails waving in the air. But the cell membrane is a little different from this. It has the double-tailed molecules, and the water is on the outside, so it's arranged like this. That was first suggested over 60 years ago, following an investigation using blood cells. It was suspected that the hemoglobin must be contained in some sort of thin membrane. Its discovery is described by Jim Metcalf.
But the realization of the structure of the membrane came from a rather simple experiment by two Dutch biochemists called Goethe and Grendel, who used solvents that dissolve fats to extract all of the fat-like material from red blood cells. Now, when they extracted the fat material, they spread it as a single monolayer at an air-water surface. And from this, they estimated the total area which all of the fat material from the cell membrane could cover. They then compared this with the area of the red blood cells that the material had been obtained from, and came to the conclusion that there was exactly twice as much area covered by the fatty material as the total area of the cell surface. And so they came to the obvious conclusion that the membrane must consist of just two molecules, a structure which was 2 molecules thick, and the only way in which this could sensibly be arranged was with the chains of the molecules pointing together at the center of the bilayer.
It was many years before anyone saw that bilayer, but look again at the electron microscope picture. There it is. The cell membrane is like a tramline. We can now actually make synthetic membranes out of good, wholesome materials. To equip it for rapid cell division, nature provides egg yolk with an abundant supply of phospholipids. To extract and study them, you need a few kitchen tools, and a little standard chemistry. After 2 stages of extraction and filtration, you get a pure solution of phospholipids, essence of membrane. Hospitals sometimes use phospholipids for their concentrated food value, injected directly into the veins of patients who can't eat or digest ordinary food. Here we're concerned with the special properties that make it the perfect material for what Alec Bangham calls liposomes. Just add water to obtain that characteristic cloudy liquid.
Well, here we've got a dispersion of membrane phospholipids in water, and if we were to look at these under extremely high power, they would look like this model that we have here. And if I move this section away, we can look inside, and the whole structure looks rather like an onion. And in between each of these layers, we have a water compartment which can contain salt or protein. And we think this as being an extremely good model for the functions of a cell membrane to act as a barrier against the outside world.
For these nerve cells, the outer membrane must be an electrical barrier too. Electricity must travel along their long, thin fibers without escaping from the sides. But at the tips, signals must pass through. At the Physiological Laboratory in Cambridge, Dennis Hayden would like to know how this works. He has a Teflon pot with a tiny hole in it and water inside and out. He paints a solution of phospholipid in oil over the hole so that it forms a thin film, rather like the bubble on the frame. Again, the colors tell of the thickness of the film. But as the oil drains away, this too turns black. Gradually, it forms a perfect bilayer, far thinner than an optical wavelength. A black lipid membrane, they call it. What's the electrical resistance of that thin black film? Hayden can check this by dropping electrodes into the salty water on the inside and outside of the Teflon pot. And he finds that the submicroscopic membrane is an excellent insulator. But now look at this. Add a certain chemical and measure the resistance again.
Okay, that's one.
Tiny signals getting through the membrane will be amplified and displayed here. Each step represents a single pore through the membrane that has momentarily opened up and then sealed again. And that's only one property of the membrane. Jim Metcalf.
Well, of course, all membranes do have to act as a permeability barrier to stop things getting across the cell membrane. But the nerve cell, in fact, is really rather specialized in that most other cells have to allow salts, nutrients, sugars, and so on to pass across the membrane in a very precisely controlled way in order for the cell to live. And it's interesting that if one goes back to the original Goethe and Grendel experiment, they arrived at the correct organization of the lipids in the bilayer because they made two mistakes which just happened to cancel each other out. They didn't know how properly to estimate the area occupied by the lipid molecules in the surface of the membrane because they didn't know how hard to squeeze them together. And so they came to the wrong conclusion for the area that the molecules occupied. But on the other hand, what they didn't know was that a substantial part of the surface of the cell contained protein molecules which turn out to be responsible for these precisely controlled passage of salts, nutrients, and sugars across the cell membrane.
Most membranes have large protein molecules associated with them. By the late 1930s, it became apparent that they must be very tightly bound, perhaps like this. But this raised a problem. Nutrients and other substances were known to pass very selectively through membranes in enormous quantities. A simple hole in the membrane looked wrong. It has the fatty tails in contact with water. So they thought of this, protein-coated pores in a rigid, protein-covered membrane. At the time, that was impossible to test.
With this rigid view of the membrane, and because techniques for looking at protein structures in membranes weren't available, there was really very little progress for the next 20 years or so.
This is one of the men who at last began to shift that 20-year logjam. Hardin McConnell and his friends at Stanford University in California devised a series of elegant experiments designed to measure not how rigid but how mobile the cell membrane is. Between the poles of a powerful magnet, they placed membrane materials mixed with a special marker that was sensitive to the magnetic field, and this would reveal any movement inside the membrane. Hardin McConnell.
Well, this is a very interesting small molecule that can be used to study biological membranes. This molecule dissolves in water above the bilayer membrane, and it also dissolves in the hydrocarbon part of the phospholipid bilayer membrane and in biological membranes. In 1968, Wayne Hubble and I discovered that this molecule was tumbling at an enormously rapid rate, roughly 1,000 million times per second. It tumbles at that rate in water also. So we had a clue that this region of bilayer membranes and biological membranes was very fluid, almost as fluid as water. We further went on to prepare other molecules. For example, this is a fatty acid hydrocarbon to which a group of this type has been attached at this position. And we have found that, first of all, the molecules have this orientation that the host phospholipid fatty acid chains have. We have discovered that the molecule rotates rapidly in this fashion. We've also discovered that the molecule translates from one site to another in a very small period of time, 1/10 of a millionth of a second. Also, this group was attached to various positions along the chain, and we discovered a flexibility gradient, namely that the bilayer is more fluid towards the center than it was near the polar head group region.
The established picture of a membrane held rigid by its protein coat was cast in serious doubt by McConnell's experiments. So how did his colleagues react?
Well, nobody believed this. This is the simple statement of fact. We obtained these results with these probes in the period around 1968, and I discussed it with various people interested in membranes, and no one believed us.
In Britain, somewhat similar work had been going on for some time. But Dennis Chapman took it a stage further. He studied the curious properties of lipid bilayers containing different combinations of phospholipids by measuring their melting temperatures. He measured how much heat is absorbed at every stage. Here it's still below body temperature, but at one particular point it's begun to absorb a great deal of heat. Something's melting. According to Dennis Chapman, it's just part of each molecule of phospholipid.
Yes, they're really rather unusual. The melting point of a typical phospholipid molecule might be something like 230 degrees centigrade. But if you look at the melting behavior more carefully, let's say you use a calorimeter where you can study the heat changes, what you find is that there's a major melting process occurs many tens of degrees below that final melting point. What essentially is happening is that at the lower temperature, the lipid chains melt. They melt separately from the rest of the molecule. The polar part of the molecule here keeps the molecule together in a crystal. So when we raise the temperature, so we go over this transition temperature where these chains melt, essentially we have a liquid part and a crystal part. So we have produced a liquid crystal. Rather unusual type of molecules.
In fact, to keep that melting within bounds, you need one further component.
There had been many discussions of the role of cholesterol, but there was considerable confusion until we try to relate this to this concept of fluidity. You see, if we have the phospholipid molecule here, and it can waggle and twist and undergo rotational isomerism motion about these carbon-carbon bonds, what we suggested was that the cholesterol molecule, being a rigid molecule, would actually inhibit some of this motion. It would, in our terms, modulate the fluidity.
A real cell membrane might look something like this, and those chunky cholesterol molecules are essential to all our lives. The old picture of a stiff, immobile membrane was dead, or it would be as soon as the problem of those proteins was solved. If they're not all over the outside of the cell, where are they? There's one clue here in red blood cells again. In the mid-1960s, Dan Branton was introduced to a new way of freezing cells into an icy block and then cleaving them with a razor blade. This is a technique devised in Switzerland with the intention of exposing cell components to the electron microscope. First, the samples are mounted on a table that's cooled by liquid nitrogen. The blade, too, is cooled, and the whole operation enclosed in a vacuum chamber. They call it freeze fracture. Ever clearer pictures to show how the insides of cells are organized. That's what they wanted originally. But red blood cells are better for studying the outer membrane. And the shape of that broken surface is examined under the electron microscope. Now at Harvard, Dan Branton recalls the arguments he had with his professor about where those fractures occurred.
It was generally believed that what we were looking at were the surfaces of membranes, whereas as I looked at the pictures and began to study them more carefully, it seemed to me evident that we were actually looking inside the membrane and that the membrane was actually being broken in two.
In 1966, Branton was in fact proposing that the lumps he saw were proteins inside the bilayer, and that the freeze fracture picture showed the cell membrane with its outer half peeled away. How was that idea received by his fellow specialists?
Well, not very well. Most people didn't believe it at the time, and it took about 4 or 5 years for me to present enough evidence to convince others that this was in fact the case.
This was one of his early pieces of evidence. The fracture cuts a section across the bilayer. As we move left, the black region opens out as a tear has pulled the bilayer apart. How safe did he feel in his interpretation of such pictures?
I certainly didn't feel all that safe. I was a young scientist at the time, just beginning my career. On the other hand, I think if one is coming up with new viewpoints, inevitably there's going to be some kinds of conflict. This was a conflict that took perhaps a little bit longer than most others that I've been involved in to settle.
It's now firmly settled, but blobs of protein in the membrane? If so, are they fixed or do they move about too? To find out, it's possible to attach fluorescent markers to certain proteins on the living cell. In an experiment of 1972 in Philadelphia, human cells were labeled in one color and mouse cells in another, and they were then persuaded to fuse together. At first, the markers are seen on opposite sides of the cell, but within half an hour, they're fully mixed together. In England, at the Molecular Biology Unit in Cambridge, high-powered researchers had come together to tackle questions about the control and organization of cells. But this man, Mark Bretcher, doesn't work too well as one of a team, so he chose a problem that his professors knew nothing about.
Well, up until 1970, no one had any experimental evidence about how the proteins were arranged with respect to the bilayer. It had been hypothesized that they were spread out on each surface, or that they were stuck inside the membrane, or anywhere, but nobody actually had any experimental evidence. And so what I decided to do was to try and find out where the proteins of the red blood cell membrane were. And the reason for working with the red blood cell is that just as its internal contents is very simple, it contains largely hemoglobin, so it would be expected that the membrane itself would be a very simple membrane compared to other cells which have much more complicated functions. So what I did was to make myself a reagent, a very radioactive reagent, which would react with the amino groups of proteins to try and tag the proteins which are, we have our bilayer here, to tag those proteins which are on the outside surface of the membrane.
First, he used intact blood, but for the second part of his experiment, he needed not the ordinary unbroken red blood cells, but what are called ghosts. To make a ghost, you mix blood with very salty water, so the cells swell up and burst. Get rid of the hemoglobin, and the tattered remnants of the membrane are accessible from both sides.
The design of the experiment then was to take the intact cells and label them from the outside and see which proteins get labeled, and take the ghosts and label them from both sides and see which proteins get labeled.
He discovered that two proteins in ghosts got labeled on both sides of the membrane.
So I was able to show that both these two proteins actually stick through the membrane.
Not like this, but more like this. The next questions are, what are those proteins made of? What do they do, and how are they anchored in the bilayer? In fact, biochemists have found there's a whole range of proteins that'll pick up colored stains even in a simple membrane like the ghost of a red blood cell.
Did it dissolve all right?
Yes, very well.
Great. And that's for the PAS?
Vincent Marchesi at Yale is using a standard method for separating proteins of different molecular weights. The two that went, as Bretcher showed, right through the membrane are similar in size. Marchesi is interested in the one that stains pink.
Well, over the last 6 or 7 years, we've been trying to work out the molecular organization of this protein. Now, what we first discovered is that this molecule is made up of 131 different units. We have determined that the units are arranged in a specific order, and the orders are represented by these symbols. The segment in the center is called hydrophobic because it's most capable of interacting with the lipid molecules. And this, we think, is the key to this molecule being able to be anchored in the membrane. Now, in order to make this point a little more clearly, I can show you right here this segment of the protein in a symbolic form. And these are so-called space-filling models. And this is the segment of protein which extends from the picture this as being the surface of the red cell. Of course, the outside part of the molecule is not shown here because it's much too large. The critical points are that this peptide, when it's sitting within the red cell, might appear coiled as an alpha helix. And this is what this model shows. If you look down here, you see that the part of the molecule inside the lipid stops here, and the rest of the chain, all the way down to the end here, the rest of this chain is probably sitting inside the intact red blood cell. Now, a crucial point in this model has to do with the question whether this stretch of polypeptide is long enough to get across the lipid of the cell. Let me show you what the dimensions would be required for a standard phospholipid bilayer. Now, these are the usual phospholipids, and I'll lay these down here so that they're in more or less the state that they would be in the intact cell. You'll notice that this segment is exactly the right distance to span the lipid regions of the membrane.
We now have one protein described completely, as a thin string with a large sugar-laden section that likes water, a tightly coiled region that water will avoid, then a tail that likes water, which solves the problem of how one of Bretsch's two proteins is anchored in the membrane. But that's too thin to show up on the freeze fracture pictures. So what are these blobs, the proteins you do see? A clue comes from an experiment in which a beam of electrons is shot through membranes to make a pattern on a photographic plate, which the molecular biologists at Cambridge can interpret to discover how another long, thin molecule has wrapped itself up. Richard Henderson and his colleague Nigel Unwin were the first to find a suitable protein and a way of probing its structure in order to produce a three-dimensional map. Level by level through the membrane, the computer charts for them a dense and regular array of protein segments. Neither Henderson nor anyone else had predicted what they saw: a pattern of thickly coiled segments suspended between one membrane surface and the other, with repeated groups of 7. Each 7 segments form a single molecule, a tightly grouped oval linked by strings that are too thin to see by Henderson's technique. This is a much smaller protein than the one in the red blood cell, but it does suggest the kind of three-dimensional layout that these larger particles may have. In fact, there's some evidence that each of them contains 2 such molecules, that transmembrane proteins sit in pairs that may work together to provide the channels into the cell. So the membrane itself probably looks like a rolling landscape of phospholipids and cholesterol, with huge boulders embedded in it. Some are proteins that make tunnels to the depths below. That mechanism is still in the realms of guesswork. Then there are the tree-like proteins with branching sugar chains above. One function of these could be recognition and identity, and below it has a root inside the cell. But what about the phospholipid bilayer itself? Does that have an inside and an outside that are different from each other? According to Mark Bretcher, it does. He made a second curious discovery when he compared his labeled ghosts with the normal unbroken red blood cells.
What I found was very surprising. That when I labelled the red blood cells from the outside of the cell, I found that none of the lipids became labelled. But then when I labelled ghosts from both sides of the membrane, then I found a lot of lipid being labelled. And what that suggested was that there weren't any amino lipids on the outside, but only on the inside surface of the membrane, and that therefore the lipid bilayer was asymmetric.
At Stanford, they'd already shown that lipids tend to stay in their own layer almost forever.
It's virtually forever. It took roughly something like 10 hours to go from one monolayer over to the other monolayer. And that time scale is to be contrasted to the time scale for lateral motion, which took of the order of a 1/10 of a millionth of a second.
In fact, the two layers can maintain their differences indefinitely. The two are completely separate. In Jim Metcalf's lab, they used detergents to dissolve away and study the membranes of real cells.
The membrane that we've mostly been working with is a membrane which occurs inside muscle cells, which is responsible for pumping calcium. Now, every time you flex your arm or muscle, these membranes pump calcium within the cell extremely rapidly, within a fraction of a second, to allow the muscles to contract and then to relax again. And what we found is that when you remove the detergent very carefully and in the right way, the membranes will spontaneously reform with the protein molecules pointing the right way to accumulate calcium. So this means that not only do the lipid molecules in the bilayer have the ability to spontaneously form the right membrane structure, but also it is built into the protein structure to insert correctly into the lipid bilayer. Another interesting thing that the protein can do is select out of the mixture of lipids in the bilayer precisely those lipids which support its function as a calcium pump most effectively. Now, we don't know how the protein can do this. any more than Mark Bretcher can tell you why the lipids on the inside of the plasma membrane are different from those on the outside. But it's clearly a very important feature of the design of the protein in enabling it to function. Maybe in the long run we can understand the significance of these structural features of the membranes by looking at how membranes work in action.
Membranes move. As they swallow material from the outside world, the membrane touches together and bites off a piece that sits for a while inside out within the cell. When hormones or waste materials are ejected, the membrane that was around them becomes a new part of the outer membrane, and that new bit was put in the right way round. What moves the membrane? In cell division, what drags the nucleus apart then draws the membrane in to fuse and nip in two. Here an answer is beginning to appear. It has long been suspected that cells have some sort of skeleton, and when fluorescent pictures like this began to appear, it was duly called a cytoskeleton. But really those strips are more like muscles. In fact, they're made of the same material. It's called actin. Beneath that first muscular system there's a second, more delicate network which seems to give the cell its shape, and yet another very fine system of filaments. It's not yet clear what these are for. When cells move over a sheet of glass, they quickly coat it with a layer of protein to which they cling. Points of attachment show as little dashes in this view, while fluorescent light illuminates muscular strands extending to each of those points. What's actually happening at one of those tiny points of attachment? To find out, you grow cells, a cloudy film of them, on a glass slip. Now fire a water cannon at it, and the cells will cling on for dear life. But inevitably in time most of them will be swept away. As they go, they'll leave traces, a sort of footprint you might call it. Now it's time for a little forensic science. A quick-setting resin will capture the print on the glass. You then chop it into thin slices. What you need is vertical cross-sections through the print. In the resin they're searching for a fragment that's quite invisible at optical magnifications, a sliced footprint. So these will go to the electron microscope. Curiously, this is the work of an industrial laboratory, of Unilever, better known for its interest in detergents and edible fats. They're doing fundamental research into cell membranes because they think that sooner or later it's going to be important to them. And the best way to hear what's going on is to have some results of your own. He sees a line, the protein that was laid down on the glass, and here is a footprint, a piece of membrane with proteins passing through attached to a further fuzzy mass inside the cell. Now explain this. A grain of dust is about to be picked up by an advancing cell, and it's carried quickly back on the surface. One possible explanation is that phospholipids are inserted in the leading edge and then flow back over the cell. An alternative idea is that the proteins themselves are pulling backwards, like this. That's the most popular picture.
I don't believe a word of it, but...
Except with Mark Bretcher.
Well, my objection to that is that if you calculate how many molecules, how many protein molecules there are in the surface of the cell membrane, there's not enough actin to go around. That is, there aren't enough... You can't have... There's not a filament for each protein, that's for sure. There aren't enough filaments. And it doesn't make any biological sense.
All good academic infighting. But when that and many other problems have been put to the test, where is all this leading to? Well, for one thing, there are many diseases, from multiple sclerosis to cancer, that intimately involve changes in cell membranes. Yet science has only just begun to reveal its nature.
Well, maybe you should try putting air in.
The cell surface is the site of many of our bodily defenses. Tiny differences may predispose one person to a particular disease and protect another. Viruses know how to break into and hide inside cells, which is why they're still so difficult to attack. So it looks as if this pure research could be very useful indeed. And there are already one or two small signs of that.
Now, as we showed earlier, this liposome model had the properties of retraining water and salt solutions inside. This property has been exploited for delivery and the slow release of drugs. For example, we can put water-soluble drugs in the water spaces here and rely on the inability of the water-soluble drugs to cross the hydrophobic membrane. Alternatively, other drugs which prefer to be in oil can be added to the phospholipids themselves, and then the slow release depends on the retardation by the water spaces.
One possible application is to arthritis. It's a difficult disease to treat, and over the years, patients have seen trials of many drugs to relieve the pain.
How long does it give you bad patches for?
Well, a few weeks at a time.
A few weeks at a time, I see. Could you just pull up your trouser leg and see how much heat it's giving off this morning? As you can see, it's still got a hot patch just to the side of the joint.
The heat camera shows the inflammation of the joint that's characteristic of an acute attack. This has proved one of the most effective drugs, but it has problems. At the Strangeways Laboratory, Cambridge, John Dingle.
Well, all the methods currently used for treating patients have disadvantages, particularly the steroids, the drugs like cortisol, which are very effective, have unpleasant side effects. And although it's still possible to treat patients directly by injecting drugs into their joints, these escape very readily. We felt that if we could encapsulate the drugs into some sort of particle which allowed them to be taken up by the very cells that we want to treat in the joint cavity, this would give us great advantages.
If it doesn't escape, far less of the powerful drug should be required. But how little and how long will a single dose last? This rabbit has a mild experimentally induced arthritis and is now being given the new treatment.
Hold it steady.
The drug has already been modified so that, used in liposomes, a single injection should continue to be effective for the duration of an acute attack, so that in the more severe human disease it'll reduce pain and limit further damage. But how can you tell?
You can't, of course, ask a rabbit as you can a patient if he feels better, therefore you will have to have some quantitative method.
So again they use a heat camera to look directly at the inflammation in the joint. They've shown first that liposomes without the drug have no effect either way. And now they demonstrate that their latest preparation does reduce joint temperature. This is over a period of days, and should keep it down for the duration of an acute attack. The properties of liposomes seem well matched to the nature of the disease.
We are fortunate we have an enclosed cavity. This cavity is bounded by the very cells that we want to treat. Therefore, when we inject particles into the joint cavity, we treat only those cells. And since those cells are very clever at taking up the particles, almost no material escapes from the joint.
But as yet, it is very early days. And that's even more the case in this second investigation.
It's a diabetic animal?
Yes, it's diabetic.
And is it a low or high dose insulin?
It's low insulin.
Yes. And it's going in orally?
Oh, yes, orally.
At the Charing Cross Hospital Medical School, Brenda Ryman's question whether insulin could be given to diabetics by mouth instead of by the usual interminable injections is just one of a range of studies of ways in which liposomes might be used in the future. They already have a way of seeing where they get to. They tag the liposomes with a radioactive marker, inject them, and then they look where that radioactivity goes before it dies away. Injected in the bloodstream, the tiny marked capsules are carried freely about the body. But what happens, or should happen, next?
It really needs to be rather like a letter bomb. It needs to be tough enough to stay in circulation. It needs to be precisely addressed so that it ends up at just those target cells that you're aiming at. It needs to be recognized and accepted by the cells that it's aimed at and taken in, and then finally it needs to blow up.
So let's try to make a letter bomb. Extract phospholipid from egg yolk and add the drug, in this case it's methotrexate, an anti-cancer agent, to produce instant liposomes. Then you pass it through a kind of sieve to separate them out, liposomes that formed with the drug trapped inside them. But will they work? Each of these rats has a tumour implanted in its flank. The aim with most anti-cancer drugs is for them to find and destroy the tumour cells without damaging normal cells which may be very similar. Since that's virtually impossible, any new treatment for cancer requires very careful testing before it can be used on man. The liposomes are mildly radioactive, so a few hours later we can see where the radioactively labelled drug has got to.
Can we try at a different saturation?
They see a strong signal from the region of the rat's liver and spleen, but nothing from the tumour. Apparently, the liposomes are too big to penetrate its fine network of blood vessels. Back in the laboratory, there's an answer for that. Ultrasonic vibration of the original mixture breaks down the big multi-walled liposomes into tiny bags surrounded by a single bilayer. Try these on a rat, and there's still a very strong signal from the liver and also from the spleen. But now there is a faint image from the tumour on the flank, and that's a start. But it does seem that they always get to the liver and spleen. The liver takes up liposomes because it has the job of breaking down such foreign materials, and the spleen gets rid of defective red blood cells. So Chris Black of the Nuffield Institute has turned his attention to a disease of those two organs. It's a major tropical killer called leishmaniasis. Spread by sandflies, its tiny protozoan parasites crawl around inside the cells of the liver and spleen. There is a drug to cure it, but that contains antimony and is itself a poison. Is there a better way? Packaged in liposomes, the drug may be a great deal less poisonous, or so the animal trials suggest. Chris Black.
Well, some of the advantages are that we should be able to use less drug to get the same amount of cure. We should in fact be able to use a single shot. In terms of tropical use, it means that a patient will come just once to a bush dispensary instead of having to come back repeatedly, maybe, for repeated shots of the drug.
So liposomes could succeed where all other treatments fail. But there are still questions to be answered.
We don't know the shelf life of the liposome in the bush, and we don't know if humans react in the same way as mice and hamsters do.
From mice to human trials, to even one successful application of this research could consume a great deal of energy and time, for so often there are hidden snags, and that's especially true in the development of new drugs. If liposomes do work for arthritis, they'll slow its advance, but to find a cure will take a lot more hard work. And for the letter bomb to find its target in other diseases will require a great deal more fundamental knowledge about the role of proteins in the membrane. The address of the letter is written in characters drawn from a vast range of different substances, about which we still have much to learn. But medicine is not the only application for membrane science. Some very surprising industries are taking an interest and have their own laboratories. Here, a phospholipid monolayer will be the first stage in the manufacture of a remarkable new kind of electrical device. A single layer of phospholipid molecules sticks to the glass. This is at the Standard Telecommunications Laboratory. Now the slide will reverse, pulling the monolayer back up to apply a second coat, and so it'll continue, layer upon layer. The result is called a multi-layer, and between or in those insulating layers you can put combinations of materials unknown to nature.
How many layers is that?
I've about 10 layers on at the moment.
Tough but biodegradable, rather like a leaf which is also packed with membranes that collect energy from light.
There's 33 to 57 in 4 layers of depth.
The multi-layers are now thick enough to interfere with light, increasing by steps to make the colours of the rainbow.
So we should go from blue right through red. almost to yellow again at the
Yeah, I
top
think
end.
you can see.
Yeah.
Only God, they say, can make a tree, but soon man will be making a new kind of multi-layer leaf that will turn light directly into other forms of energy. So standard telecommunications are into biochemistry, and imperial chemical industries are into electronics. Here, a silicon semiconductor has been coated with insulating layers of lipids. ICI's Bill Barlow.
If it is possible to produce a device such as a transistor, which can incorporate a whole series of antibodies or antigens, it may well be possible to diagnose a whole range of various problems of the body by a one-shot sample. Now clearly, if this is possible, then the implications to medical diagnostics are enormous.
What they're aiming towards is a transistor composed entirely of biological materials. That's as fundamental and potentially as far-reaching as the invention of the transistor itself. In the final stage of making this biotransistor, they have to add an electrically conducting layer by condensing a film of metallic gold onto its surface. To protect the lipid layers, that has to be done cold. They use liquid nitrogen. The new technology may take another 10 or 20 years to develop and then to apply in ways that now seem far outside the realms of practical science. For example, to detect and identify single molecules. British industry has a few laboratories like this. In America, too, industry is reported to be on the move. Firms like General Electric and Union Carbide, and interest is growing fast. Jim Metcalf?
Well, I suspect that in the next 10 to 20 years, somebody is going to make a great deal of money out of the technical development of membranes. But in the end, this will depend on the very pure basic research which has been carried out in the past 10 years or so, guided simply by the principle of people looking where the light shines brightest.